All right, this is in the class, and I'm going to have you watch a movie for that particular quiz. You'll watch the movie. I'll tell you what to look for in the movie for the quiz, or you'll find it. You'll watch the movie and do the quiz while you're watching the movie. That'll work. But it's a documentary, and it's pretty good as far as documentaries go. It keeps you engaged, anyhow. It's not dry at all. Um, but before you see that movie, I kind of want to set up the idea that just how important the promoter is to the future of biological research, and especially diseases and medicine and that sort of thing. The one thing I want you to realize that we've been looking at the protein coding region of genes for 50 years, since the 60s. Um, but just recently have we started looking at the promoter. And we only did that after we started sequencing the human genome and then getting good at it and sequencing many genomes. And here is an early paper that shows the recognition that, hey, there's a lot going on here, and we haven't been looking at things correctly. This is way back in 2009, and the Human Genome Project really is just now coming to the point where we're finished. They're finishing up the last few bits. It's not sequenced well here, but in a year or two they will have a really good sequence, including the parts that are not protein coding. In other words, the promoter areas are the region that we're talking about. And here someone reports back in 2009, which was a big deal back then to report something like this, that there is a growth factor. So a growth factor is something that is probably, you would think of it as growth hormone or something. This is a gene that is related to your cells dividing and your body developing and growing into like an adult, or either repairing wounds. There are lots of different types of growth factors. But here there is one called TGFB1. So transforming growth factor B1. This is probably a protein that binds in the promoter region of many genes. So in other words, we're going to call this, and this is related to the movie that you'll watch, a boss gene. So keep that in mind. This is the idea of a boss gene. So I'm going to just write it here. A boss gene. All right? Right there. A boss gene, capitalized BOS. In other words, it bosses other genes around. And that's what transcription factors, or the proteins that bind in the promoter, do. They boss a gene around. And realize many promoters have the same sequences, and these proteins, like TGFB1, would boss them around. And it's a growth factor, so you assume it means, hey, go make more of whatever you're making. All right? So it would bind to the promoter region, all right, of a gene at these sequences that we've been studying here at the end of the course, and it would attach and increase the rate of transcription. So you could think of it as an enhancer, or there are lots of different terms to use for it, but it would increase the rate of transcription. So you're going to make more mRNAs, and you're going to make more of the protein, and we assume this is going to cause the cells to multiply. So that's what transforming growth factor beta 1 does. Transforming growth factor TGF1. And it's going to bind in the promoter region of many, many genes. All right? Not just one or two or even a dozen. Probably in 1,000 or more genes it's going to stick. So it's a very important gene. Not all genes are equal in their importance. Now, what this paper finds is that they found that there is regulatory variation in the TFB gene. Now, what does regulatory variation? It means that the promoter of this gene has some variation in it. In other words, the protein produced by this gene binds in front of the promoters, or in the promoters of many genes. But it has its own promoter as well. And what they did, they looked and they found 9 promoter polymorphisms. Polymorphism being a mutation. In other words, the sequences were changed. And they have identified a 2,000 kilobyte region upstream of the transcription start site. So we understand that in this class. We get that. They were looking at the promoter, the pre-promoter, and even the distal promoter, perhaps. All right, with 2,000 base pair ahead of it. And they found 9 distinct promoter haplotypes. Well, haplotypes means different versions of a gene. So here they're using the term correctly. They're treating the promoter as part of the gene. Now, if you read a typical undergraduate textbook, whether it's a freshman or even a genetics textbook, they're not going to use this type of terminology. This is one of the first cases I've found where they actually treat the promoter as part of the gene. And they do it by calling it a haplotype. That means you get a mutation in it, you get a different phenotype. Phenotype being what the organism looks like or how it behaves. And so they're saying that they have 9 distinct promoter haplotypes, mutations in the promoter. Think of it that way. Some people have green eyes, some people have brown eyes, some people have blue eyes. Well, those are all haplotypes, all right, for eye color. Well, they found 9 of these caused just in the promoter region. And in fact, this is exactly what we find more of now. This was one of the first cases of this, and they found 5 distinct continental population groups. So continental means Europe. So they were looking at Europe. And so they found 5 groups, and they looked at these people, and they looked for where there were mutations. These mutations were not big. They didn't cut out a big piece of the promoter. They were literally single bases. So here, here, here. Well, here's one with 3. But the idea here is they're showing us that it doesn't take a lot to alter the promoter in a major gene, one of this BOS gene. All right, so if we call this thing a BOS gene, we didn't alter the BOS gene. Okay, this paper, okay, does not, all right, alter it in the normal way. It does not alter the protein produced. All right. What it's going to do is instead here, this BOS gene that controls many other genes, its own promoter was altered, so we make more or less of the BOS gene. All right, you assume. And that produced distinct phenotypes, distinct versions of these organisms. We're talking humans here. So in other words, somebody had longer arms, somebody had bluer eyes, someone had, bigger muscles, something like that. All of this, all right, something happened. There was distinct phenotype changes. In other words, you could tell these people were different, and they were all based on very simple changes in one gene, like one base change in a BOS gene, a gene that bosses other genes around. That makes a huge difference in the organism, all right? It's not a change in the protein, all right? This is a change in just how much of that protein is there and how much of it then is available to go stick in front of other genes and regulate them. And so this is a big deal. This is showing that there are major differences in how an organism can develop, and we've talked about development in a video and differentiation, and that's where this would normally occur. This would be a gene that kind of bosses them other genes around, and probably during development in the womb, or maybe at puberty, something like that. And you alter how much of this is made, and of course you alter the complete structure of this organism with just a single, like, one base change in the right spot. So in other words, it doesn't take a lot, all right? And when you're altering the promoter. In fact, we now believe that the majority of the differences between us and between other species have to do with differences here in the promoter. And it doesn't take much, all right, to do this. And we now are seeing that here they show that these mutations could be identified as pathogenic. In other words, diseases. In other words, they're going to talk about this in here, about how these mutations not only were haplotypes, but they also can result in disease, in pathogenesis. In other words, disease. You get a disease when you get one of these mutations. This means this gene is way more important than most other genes. All right, this is a boss gene, and we're going to use that term here. It's a transcription factor. In other words, or I should say, it's a protein that can bind to the promoter of other genes, which defines it as a transcription factor. But then there are some transcription factors that bind in front of other transcription factors. And there are these major ones that really control many things in your body. And you get any kind of mutations in the protein or in the promoter region, all right, in the protein coding region, and if you get any kind of mutation in those, you probably kill the organism. They're not as important as ribosomal genes. Pretty much ribosomal genes cannot be altered in any way, or you end up not surviving to birth. But in fact, you probably don't even develop past 2 cells or something. You just don't change ribosomal genes. But the idea here is not all genes are created equal, and the genes that actually bind to the promoter region, which we're calling transcription factors, are more important than your regular protein coding genes. And then the transcription factors that control other transcription factors are even more important than those. Those are sometimes called, they're, depending on what they do, they might be several names, but a frontier factor. Those would be the ones that are in the womb. There are others, though, things like this one, growth hormone type factors, that, they are more important, and you just cannot alter them without getting major changes in the organism. Now, this is turning most of medicine and genetics on its head. And in fact, you still don't get this taught at an undergraduate level very often. Someone who's teaching molecular genetics, not molecular biology, but molecular genetics, would mention this. But, it's going to be glossed over in other courses because it's just not that well, I don't know, what do you want to call it? It's not that well known. I mean, I think most professors know it. It just doesn't make it into the textbook. And when it doesn't make it into the textbook, the professors tend to not teach it. But we don't have a textbook in this, so I'm free to teach this, and I definitely think this is worth knowing. And we know a lot about this. We know that this is true, and we probably need to be saying out there that the genes, what we used to call a gene, all right, the protein coding part, all right, the reading frame, all right, isn't all that important. It is, but I'm exaggerating there a little bit. You mutate this, and if the protein doesn't work right, your cells die. So these don't mutate that much. Most diseases, what I want to point out is, most diseases are caused by mutations. All right? Not mutations, mutations. All right? In the promoter. Okay? If you get a mutation in the reading frame, all right, a lot of times that protein doesn't fold correctly, it doesn't work, and you never make it past one cell. You are a 1 cell or 2 cell or 20 cell organism and you die, all right? You die in the womb. But you get a mutation here, okay, you make the wrong amount. Probably you're not working optimally, your cells aren't, but they're surviving. And so you actually make it to birth, but you have a disease. And most of the diseases we are learning, especially modern diseases like, let's say, diabetes. There we go. And Alzheimer's, things like that. See if I can spell Alzheimer's. I don't think I can. Yeah, I didn't spell it right. So I'll say ALZ abbreviation. That'll get me around it. All right. So diabetes, Alzheimer's, anything metabolic. Usually, you don't digest something correctly. You have anything where you have to regulate your blood sugar, like diabetes. That defines diabetes, actually, including also things in your brain, things like what was it I think about? ADHD, maybe. Things like schizophrenia. So we'll just say etc. there. Those are all being linked to mutations in promoters, not mutations in the reading frame. All right, and we just don't know enough. You're beginning to see how little we know. We know a few transcription factors here and there. Maybe we can identify where they're at in the sequence, but we're not certain. You can see how little, we're still learning. We haven't found all the transcription factors. Sometimes we have found the transcription factors, but we haven't found out where they stick. And then in other cases, and this is in humans, this is not in something like EHUX or something, we still have things to learn. Then we have sequences in the promoters that look like they should be transcription factor binding sites, but we don't know what protein sticks there. For whatever reason, we can't get it to work in the lab. So we don't know what actually sticks there. So there are so many things, and we didn't even get into RNA interference, which is important. And then we haven't talked too much about histones. We know a little bit about how they work. There's a lot more to that. Maybe Dr. Houston will go over more of that in, especially the histones themselves can be methylated and acylated. Now, we know about CPG islands being methylated, but realize you can modify the histones themselves so that they are more or less sticky as well. So it's not just the CPG islands that get sticky, but the histones themselves can essentially become more or less sticky. And that's a whole nother part of how we turn on and off genes and when, and whether or not RNA polymerase can attach at different times. And then there are different types of histones. So all kinds of crazy things that we, of course, didn't get a chance to talk about in this course. But all of those things are turning out to be far more important than the mutations in the reading frame. And so if you go to something like 23andMe or what's the other one, where they sequence your genome and they tell you the mutations you have, most of those mutations are in the reading frame. And those are not most of the diseases out there. And they're just the ones that we knew about before we realized that the promoter was actually the more important part, because we didn't know how the promoters worked. Uh, we knew they were there. They were probably important. We didn't realize how important. And what I'm going to have you do is watch a movie. So there is a movie I'm going to link to this video called What Darwin Never Knew. All right. And that is going to be, it's on YouTube, it's free. And it's one of my favorite documentaries. And it's made at a time where they're just now discovering that promoters and transcription factors, those are really important things, but nobody had ever altered one to see if it did anything. And in the movie, the latter part of the movie, someone does that. They do it with flies. So you'll need to know that story for the quiz. So there are a couple of things I want you to know from the movie. I'll give you a link to the movie in the announcement. But besides watching the movie and listening to this, I want you to be able to tell me certain things from the movie in that quiz. So when you're watching it, be ready to pay attention to these things. And one of them, I want you to realize they call something a switch. All right? And a switch, just think of it as a transcription factor binding site. So one of those transcription factors binds at a sequence, and they're going to be altering that switch. So a switch is, in the movie, is called, so they try to keep it simple, and so they call it a switch, but it is a transcription factor binding site. So we've talked about one, GAF. That would be the transcription factor, and it binds right at GA, one of the easier ones. All right, so that would be an example. So keep this in mind when you're watching the movie. That's what they're calling a switch. Now they also have something, the transcription factor itself is, they're going to call it a boss gene. Now I've just explained that there are different levels of transcription factors, and there are boss genes that boss around other transcription factors. So realize, but their boss gene is essentially an important transcription factor. Now, they don't go into what we do, all right? They talk about more of the evolutionary implications of what is going on, all right? Now, the scary thing, this is before CRISPR was invented, so it was a lot more difficult to alter these things. These days, we can go mess with this relatively easy. I didn't say it'd be easy, but we can go and alter these, and people are doing what they're doing in this movie pretty easy. They did it the old-fashioned way before CRISPR. It was pretty hard. All right, so what they're going to do is they're going to alter a fly, and they're going to make its spots appear and disappear. And so pay attention to that. There's another part that you're going to be quizzed on about the movie. There are 4 researchers at the end, and I want you to be able to tell me what they did. Researchers at the end of the movie, last 30 minutes. It's about an hour and a half, hour 50 minutes. All right, so the 4 researchers, and you'll know who I'm talking about. There's going to be a guy who studies thumbs. Very important for humans. Thumbs are a big deal. All right, another one that studies muscles. All right, another guy that studies children's brains. All right, and then the last lady, she is the bioinformaticist. All right, the bioinformatics. All right, so she does what I do. All right, in the at the end, and she shows some very important things that I want you to know. So pay attention when you get to the last 30, 35 minutes. And you'll start with the thumb guy, and I think the muscle guy comes next. So I don't care if you know their names, you can just remember them by these terms. What I want you to be able to do is tell me what they found. What did they discover? Why was their discovery so important? All right, so I want that, and then I want you to understand how hard it was for them to do this without CRISPR. I'd like you to understand that, and put it in perspective to evolution. All right, keep in mind that's what most of this is. It's what Darwin never knew. Darwin never knew about promoters. All right, so there are things that he could have never understood, because one, he didn't even know DNA existed and how transcription and translation worked. But more than that, we now know more than we did just 20 years ago. There's none of what we're doing now was even suspected really 20 years ago. So watch this one. This movie kind of puts things in perspective, and why bioinformatics is dominating not just medicine and things like that, but it's dominating evolutionary biology, and we're changing the way we think about how organisms are related, etc. All right, so please watch this movie, watch this video here, and then go watch the movie at some point before, sometime next week. Whenever I put the quiz out, probably the best way to do it is to watch it when I give you the quiz. But it's actually an entertaining show, so you could watch it. I mean, I don't know how much time you have. You probably don't with finals coming up, but keep in mind that I will quiz you on this, and you're going to have to watch this thing. All right, and it's really one of the main things I want to get across in this course. All right,